Dr. Carolina Arias has graciously offered to present a seminar that's very timely. And in fact, since Carolina made this slide, I'll give her title, A New Virus on the Block, SARS-CoV-2, which is the name of the virus, and the COVID-19, which is the name of the disease, epidemic, which is now a pandemic. And let me tell you a little bit about Dr. Arias. She's an assistant professor in molecular, cellular, and developmental biology at UC Santa Barbara. And we're really fortunate that she's done this on really short notice. She earned a PhD in microbiology at NYU and the Sackler Institute in 2008, where she worked on herpes and pox viruses. And as a postdoc, she worked in Don Gannon's lab. Many of us know about that lab at UC San Francisco, and then went to Novartis Institute. She also joined Joe DeRisi's lab at UCSF just before she came to UCSB. And we welcome Dr. Arias and look forward to hearing what she has to say about this very important pandemic. Carolina.
Okay, well, thank you, David, and thank you everybody for being so flexible and for joining this meeting remotely. This is not only a great opportunity for me to share with you some of the passion I have with for viruses and virology in general, but it's also a great opportunity to start testing these ways of meetings and these ways of not only teaching but also sharing science and sharing information remotely. So this, I hope, is going to be technologically sufficient and successful, and I will look forward to some of your comments in that regard. But I think all of us are aware of the situation that is going on. We're constantly bombarded by information in the media, and I just thought that it was going to be a great opportunity for us to have an update on what is going on from the point of view of us, the virologists. So I'm going to stop sharing my video right now. We're going to focus on the slides, so just bear with me for a moment until I figure that out. Give me 1 second. Good. Okay, so as I said, one of the things that we are seeing constantly in the media is all this information about COVID-19, and is as of today being upgraded as a pandemic. This is a clip from the news that I took from just a month ago, where we had just named the disease COVID-19. So this just shows you how rapidly the situation is evolving. But one of the things that I want to remind you is that this is not the only virus that we have seen in the news, and this is not the only outbreak or epidemic that has plagued the world in the last years. We are right now living through a flu pandemic, or a flu epidemic actually in the United States, that has also been pretty debilitating and has affected a lot of people and has caused a lot of suffering and death. So this is something that we should also be aware in the background of this current epidemic. We have also had a lot of outbreaks in the United States of measles. This disease was almost eradicated, but thanks to the anti-vaccine movement, the disease has come back and has come back with a vengeance. I'm going to show you an example of an outbreak that we had not so long ago and not so far from here of measles. Of course, many of you have also heard and are aware of the Ebola outbreaks that has been declared in the Congo constantly over the past 3 to 10 years. We've seen a lot of these epidemics coming and going. Again, this is a disease that causes tremendous suffering in the populations that are afflicted. And one of the diseases that we study in the lab, which is the Zika virus, which caused a major epidemic between 2015 and 2017, where mothers that were exposed to the virus during pregnancy transmitted in utero to the developing baby, and these infants were born with severe malformations and problems, including microcephaly. So this is just an example of the kinds of viruses that we are seeing in the news and the outbreaks that we, again, are constantly facing. There are more, but now that I painted a very dire situation, I'm here to tell you, do not panic. This is taken from one of my favorite books, which is The Hitchhiker's Guide to the Galaxy, and many of you are familiar with this message. Do not panic. That also brings me to, this is one of my favorite quotes. I know that this COVID-19 situation is bringing a lot of anxiety for many of us, and in part is because we know so little about it. So this quote by Marie Curie, which is nothing in life is to be feared, is only to be understood. And now is the time to understand more so that we may fear less. And I couldn't find something more timely and more applicable to this disease than this quote. We are afraid of what's going on because we don't understand it, but we are making a lot of effort to try to understand the current epidemic, how is it developing, and how is this virus causing disease. So just to get familiar with some of the topics that I'm gonna be discussing today, one of the first things I'm gonna tell you is, what is the SARS coronavirus 2? This is the causing agent of the COVID-19 epidemic, and we're going to discuss where it came from, what kind of virus it is, and what we know about it. We also are going to talk about where the disease came from. Sorry. Where the disease came from. We're going to discuss a little bit of why COVID-19 is just not another cold. And then we're going to talk about how can we prevent it. Give me 1 second. Okay. So let's just make a pause for a moment. I think my phone was ringing, but one thing that I wanted to mention is, if you have questions, please submit them via chat. So right now we have a lot of people online, so it's going to be hard to handle live questions. So there is a chat button that you guys can press and just submit the question. I'm going to address them in a timely manner, either during the talk, or if it's convenient, or at the end of the talk. I save some time so that we can discuss some of these questions. Okay, so before we get into the matters of what is the coronavirus that causes this disease, I want to bring you up to speed with some terms that we have heard in the news a lot. So one of those is an outbreak, and that is defined as the case. Sometimes we have an uprise in the number of disease cases that are diagnosed in a specific geographic area. So again, we refer to that as an outbreak, and that's how this disease has been called for a long time. You know, the COVID-19 outbreak or epidemic. And we'll talk about what an epidemic is. Just to put it in the context, not so long ago, last year, around this time, we had a measles outbreak. It was in April of last year. We had a measles outbreak in LA. And in this case, what happened was, one person who was diagnosed with measles went to school during the time that he was contagious, he or she, I don't remember, and this person was contagious and exposed about 500 people to measles. All these people were contacted, and 400 of them were able to show that they had an up-to-date vaccination record. 100 of them were not and had to be quarantined for a couple of weeks. Fortunately, since measures were taken to contain people, the outbreak was contained and it never evolved beyond that. And we're having this kind of measles outbreaks all throughout the country, which it's pretty sad to hear. So in many cases, what can happen is that outbreaks can expand and can get out of control. And that's when we start referring to these diseases as epidemics or pandemics. An epidemic you can consider an event where you have active spreading in a geographic region. And it's basically an outbreak that got out of control. So this measles outbreak that I told you was pretty limited. So it was not throughout all LA, but it was just in a very confined region. Once, if it had gone out of control, it would have been considered an epidemic. The COVID-19 was an epidemic up to yesterday, actually this morning at 9 a.m. in the morning, it was declared a pandemic. What that means is that the epidemic became widely spread throughout the world. So basically now we have active spreading in different regions of the world. And right now it's in every continent except Antarctica. So this is the status of the situation. So this was the announcement of the WHO, where they declared it a pandemic, which in some of our opinions it was about to happen. This is the map of the disease that we had 2 days ago. So I've been trying to keep these slides up to date. So this number of cases is probably a little bit higher. This is from either today morning or yesterday morning. In any case, as you can see, all the orange dots here are representing areas of active spreading and active disease. And again, some of the numbers of the cases that are having diagnosed that already are outdated. But this just shows the magnitude of the disease and the magnitude of this epidemic. Okay, so what are the coronaviruses and basically who is causing this disease? So let's move a little bit into the part of virology. So these are positive strand RNA viruses that the structure is shown. Let me see if I cannot see my, okay. The structure is shown here. So they have about a 30 kb positive strand RNA that is surrounded by, you see, an envelope. And in that envelope, we have glycoproteins that are protruding, those proteins that you see there in yellow. The structure of those proteins is distributed in such a way that when we look at these, at the structure of the viruses by electron microscopy, they resemble a crown. And that's where the name comes, corona. So these viruses are, as many coronaviruses, is a large family of viruses, and many of them are associated with mild disease. They are commonly isolated from upper respiratory diseases, and they cause a common cold. Something that many people are not aware of is that they are responsible for about 30% of the cases of common colds in humans. So these are viruses that circulate among the population and are common. However, they were not considered highly pathogenic until 2002 or 2003 when the situation changed. What we had at that moment was the appearance of some coronaviruses that were causing severe disease. And many of you are familiar with these images from 2002, where we had the SARS epidemic. SARS stands for the Severe Acute Respiratory Syndrome. It was the first severe coronavirus that was identified in China. We're going to talk a little bit about the origin and how it was, the transmission was identified. It was a deadly disease, so it had about around 10 to 12% mortality. There were 8,000 cases that were diagnosed around the world. And since then, there's been sporadic cases, but the epidemic was controlled in a timely manner. In part, it was because the majority of patients were contagious once they exhibited symptoms, and it was transmitted mainly in hospital settings. So it was easy to contain and control it. About 10 years later, after the SARS epidemic, we had the discovery of the Middle East Respiratory Syndrome. This one was even more deadly than SARS, so it caused about 30% fatality rate. We had about 2,000 cases. It never reached the level of pandemic because given the high mortality, it was contained pretty aggressively. And the same as SARS, it was also transmitted mainly in hospital settings. So we've had, again, SARS in 2002, MERS in 2012, 2013, and of course, right now we have the epidemic in China. So all these images are just very reminiscent of what we were seeing with both of the previous pandemics. Here, and I'm going to emphasize that later again, it's important to remember that the COVID-19 does not have a mortality rate as high as that one of SARS or MERS. Okay, so what is the virus that causes this COVID-19? So I already told you about SARS and MERS, but who is causing COVID? So you have probably heard all of these names in the media. So it's been called the Wuhan virus, it's been called the novel coronavirus 2019, it's been called the COVID-19 virus, but finally, after I think probably a month and a half of the deliberation, virologists finally came to a consensus, which sometimes is very difficult when we are naming things. But the name that we came up with, the accepted name is now Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. So the name, of course, is based on the pathology that this virus is causing, but it's also alluding to the relationship of this virus with SARS. So if we look at the relationship at the level of genome conservation, and this is likelihood tree that we're seeing here of relationship of these viruses. So we see in red, we have the human coronaviruses that have caused severe symptoms. So in the upper part, you see SARS-CoV-2, which is related to SARS-CoV, so to the virus that causes SARS. However, it's important to make the distinction here that right now there is no evidence to suggest that this new strain, the SARS-CoV-2, evolved from the original SARS. This is an independent event, and this is an independent virus. So we can consider them almost cousins, but they are not one evolving from the other. You also see that they are more distant from the MERS coronavirus, and they are definitely separated. So they are in a different branch of this tree from the viruses that cause the common cold that are highlighted in yellow at the bottom of the tree. So this is just to show you that all of these viruses are related to each other, but at the same time, genetically, they are very different. One thing that I want to bring your attention is right below the SARS coronavirus in red, you see that there is a bat coronavirus. And the reason is because these viruses circulate in bats, and that is an important fact for what I'm going to tell you next. So we know that coronaviruses can be spread by animals. So I just told you that there are the coronaviruses that cause the common cold that are more closely related to each other, and those that cause severe disease, SARS, MERS, and SARS-CoV-2 that cause severe symptoms. The common cold syndromes can be transmitted person to person, and those are the ones right here in the left where you see the two people shaking hands, which is what we should not be doing and avoid at all costs right now. But they transmit just like a common cold, right? So they are just, imagine somebody's sneezing, somebody is coughing, somebody blow their nose and didn't wash their hands, you're gonna be exposed to these viruses. The transmission of the severe diseases is a little different. It can start from animal to person and then from person to person. So we know, for example, for SARS, the virus circulates in bats, and we're going to talk about white bats in a moment. But we know that there are coronaviruses that are circulating in bats. SARS is one of them. And then we know that somehow it was transmitted to civet cats, which you see there in orange at the bottom of the image. These civet cats are wild animals that are sometimes consumed by some populations. And we know that during the outbreak of SARS, there was exposure or human exposure to SARS from these civet cats. So it was easy to track, and then these animals have been prohibited for human consumption. So basically they are banned from the market, but they can be still found in black markets. And this was one of the things that they found in Wuhan. For MERS, we know that they also circulate in bats, but in this case, the intermediate host is dromedary camels. So when you look at the pattern of this epidemic, the most afflicted people, or the people most affected by MERS were adult males. And that is just cultural because they are the most exposed to dromedary camels because they take care of them. So this was active exposure to an infected animal, and then they acquired the virus there. For the new coronavirus, however, we still don't know where it came from. And that helps me transition to the next, which is, where is this virus coming from? So what happened? How did we get exposed? And where is it? So the whole epidemic, as probably all of you know, started in Wuhan, in the Hubei province in central China. So they were able to track the origin of this outbreak to a wildlife market in Wuhan. So quickly, there was the thought of, okay, this is another SARS, and there are civet cats here, and it's very likely that they are contaminated. The civet cats that were found in the market, although they were banned, they were still some there, they were tested, and they did not carry the new strain of the virus, or the new SARS-CoV-2. So then the wild chase for who is intermediate host started. And the reason why there's so much attention and so much emphasis in trying to identify the reservoir and the immediate host is because once we know where they come from, we can control them better. So, example, with SARS, we knew that they were coming from these cats, they were banned, and then the population were not as exposed. In this case, we have no idea where it's coming from. But as you can imagine, there was a lot of attention and there was a lot of effort trying to identify it, and then there was a lot of confusion. So this was just one of the many headlines that you can find on that topic. Bad snakes or pangolins inside of the hunt for the animal behind coronavirus outbreak. And I can tell you that yes, they are in bats, as I'm going to show you. No, they are not in, or there are some coronaviruses in snakes, but this is not coming from snakes, and definitely not from pangolins. And I want to focus for a moment in the example of the pangolins. So there were so many news outlets that said the pangolins are the cause, right, or the intermediate host. So they have a virus that is 99% similar to the SARS-CoV-2, so for sure it came from there, and it was published. Well, it turned out that there was a miscommunication between the bioinformaticians and the scientists. And there is one protein that these two viruses, the coronavirus from the pangolins and the coronaviruses from humans, share. And that protein is 99% homologous or identical. However, the rest of the genome is less than 90% homologous. That means that these two viruses are not the same. And it just happened to be that there is a virus in pangolins that is similar, but distantly related to this new one. So today, we don't know what is the intermediate host, and there is, again, as I said, a lot of attention and a lot of active research into what is the animal that facilitated the transmission of the virus to humans. One thing that we know for sure is that it came from bats. So there is a bat coronavirus that is 96% homologous to the strain that is circulating in humans. So what is thought is that this virus in bats reached an intermediate host, it mutated, which would probably account for that close to 4% in variability, and then it was transmitted to humans, and it's very likely that within the human population it also mutated, as we know it's currently happening. But for sure, it's coming from bats. So that, it's pretty interesting, and I was discussing with some of my students the other day that bats are incredible reservoirs for a lot of viruses that are causing very severe pathologies. So they can carry Ebola, or I think at least we think that they can carry Ebola. Again, we have not found the circulating strains of Ebola in humans in bats, but we know that there are some that are closely related to the ones that affect humans. We have Hendra, we have Marburg, we have Nipah virus, which is also a terrible disease. We have SARS, and we have now this SARS-CoV-2. So again, one of my students the other day asked me, like, but why, right? Why are these bats carrying all these viruses? And the interesting fact is they do not get sick, right? So there's been a lot of attention into this fact because, well, first I should make the disclaimer that we should not go hunt bats and kill them all, because they're actually very important pollinators and they are very important for the ecology. But we just need to understand what is different about them, and maybe that would also help us understand how could we control some of these viruses, right? So there are a couple of explanations. So one of those is that bats can respond to viruses and mount an immune response, but they have a thwarted inflammatory response. So there is a repressor element, a transcriptional repressor element in the promoter of TNF-alpha. And it is considered that this repressor is what is reducing the response of that promoter in the case of an infection. And so when you delete that repressor, the inflammation increases and then you have problems, right? But so basically here is, unlike in humans that our immune response is going to trigger a difficult inflammatory response, difficult to control, in the bats, that inflammatory response and the damage that is caused as consequence of that is reduced. It is also thought that these differences in the inflammatory responses and in the immune responses of viruses came because of the evolution or as a consequence of the evolution of flight. And one of the consequences of flight is the elevated temperature. So these bats, when they fly, I think they reach temperatures above 38, 39, something like that. And so the viruses that are going to reproduce inside of the bat have to be able to support or to tolerate those temperatures. And that is a complication when those viruses are transmitted into humans, because a human can develop a fever, and then usually that's a response of your body to try to eliminate the virus. But in this case, that response is not going to work as well with the viruses that have adapted to live at higher temperatures. So it's just a pretty interesting, evolutionary complication. And again, the bats are not benefiting at all from transmitting the virus to us. This is just an accident of us basically invading some of their space and becoming more or coming into more close contact to them. And I put a link there to a YouTube video where they explain some of the ideas that are behind the resistance of these bats to the disease caused by this virus. But I think it's a very interesting question. Okay, so now let's move a little bit more into the disease itself and how the COVID-19 pandemic evolved. So most of us are aware that this disease started appearing into the media limelight around December 31st or the end of December of last year. So that's when the first cases were diagnosed. But it is considered now, knowing more of the epidemiology and the progression of the disease, that it's very likely that this virus appeared somewhere in early to mid-December of 2019. We know that, again, around December 31st, the first patients with pneumonia of unknown etiology were diagnosed in Wuhan. And once there was no agent to blame for the pneumonia of these patients, samples were taken and the sequencing of the genome of the, or the identification of the pathogens by genome sequencing took place. We know that the genomes, I think, came out probably like a week later, around the first days of January. We had the first sequences where a new pathogen was identified as the 2019 novel coronavirus. The 7th of January, there was the first isolation of the virus from a patient with pneumonia. And then by January 11, the cases rise from a handful to about 41, with the first mortality reported and the first genomes sequences were released. So for me, that was just, it's an amazing effort in the scientific community to go from a disease that had no agent to be diagnosed to an identified microorganism and the genome sequence within 11 days. In January 2021, we had the first cases confirmed in the US, and in January 30th, the WHO declared an international public health emergency. So this is a graph of the evolution of cases. So I just told you that the first cases in the US were diagnosed around January, or the end of January. But we can see here in yellow how the number of cases increased dramatically from the end of January to the first week of February. And we went again from a couple hundred to tens of thousands. This was in part because of diagnosis, awareness, and then because of clinical diagnosis. So there were some spikes in the graph where you could see that besides the disease being diagnosed by testing, it was also diagnosed by symptomatology. You can also see that around between the 16th of February and the 7th of March, the yellow line plateaued completely. And this is the outcome of the quarantine and the isolation, social distancing and isolation measures that were taken in China. And these basically prevented the epidemic from expanding. And now it is being reported that many less cases are being diagnosed in China than around the world. Not only the diagnosis decreased, but also, of course, the death was decreasing. But the reported patients that were reaching hospitals because of respiratory symptoms dropped more than tenfold. So when the epidemic was rampaging, the people were going by the hundreds to hospitals, and now there's about 50 people that go look for care every day. So the situation in China is looking promising and is no, I don't want to say under control, but definitely is looking very, very good. That cannot be said though from the cases outside of China. In the blue line, if you see the speed of diagnosis is basically mimicking what we had in the initial stages of the cases in China. So it's very likely that we are going to hear of more cases being diagnosed and these numbers quickly rising outside of the world, as we have seen in the past few days. Okay, let's take a look at what is going on in the US. So again, in January 2021 was the first case announced in the US. We knew that in Washington, and it was possible to trace back the origin of this case. So this was a person that was known to be exposed to somebody who was diagnosed. So you could trace back this case to a known source. On January 30th, as I say, the WHO declared the global health emergency with more than 9,000 cases reported worldwide. In February 8th, the first US citizen died from COVID in Wuhan. And in February 26th, California announced the first case in the US with no clear source of exposure, which is where things got a bit more worrisome, because we could not track this case, or the authorities could not trace back this case to a known person that had been diagnosed. So what this implies is that there has to be, or must be, a community-acquired case. So this virus was circulating somewhere in the community where this person got exposed and acquired the virus. In February 29, we had the first case of death, a person in Washington State. In March 6, Trump signed the $3.8 billion emergency spending package, which, if you see the time distance between January 21st and March 6th is a measure that had to come at some point. And it's unfortunate that we had to wait for the global cases to hit 100,000 to declare these. And in March 6th, so just a few days ago, the Vice President announced that 21 people in the cruise ship, in the Grand Princess, tested positive for the coronavirus, and now we are in the midst of this disaster with the cruise ships that they were going to dock in Auckland, dock in Santa Barbara. We don't know when they're docking, they don't know when they're getting off. It's just a very dire situation for the people that are on board these ships. Okay, so this is how the situation looks as of yesterday, according to the CDC. So this is the distribution of the virus across the United States, and as we know, California is one of the places where we have the most cases, together with Washington and New York. It's not that surprising, since we have a big number of large cities in these states. These numbers are constantly changing, so apologies if they are already outdated. I updated them yesterday, so I am sure they are already wrong. But in the US, we have more than 1,000 cases, 31 deaths, and 15 cases known that are recovered. But again, these are the cases that are reported to the CDC, and it's possible that we have some more. Okay, so let's talk about now about the disease. So this is the list of symptoms that has been reported by the CDC. And the symptoms are very non-descriptive. They include fever, cough, and shortness of breath. And if anybody has had the flu in the past weeks, these are the symptoms of flu as well. So that's a confounding factor that we have, because many of the symptoms are just common, and it's going to be very hard to diagnose. We don't have an accurate test that is going to allow us to differentiate between one or the other. So we can test for influenza, and the tests for COVID-19 are coming. We know that the symptoms, or the CDC reports that the symptoms come between 2 and 14 days after exposure. And this is not only based on what is seen in this epidemic, but it's also with what has been previously reported as the incubation period for MERS and SARS. The complications that are seen in some more susceptible populations are trouble breathing and pneumonia, which is defined an infection that causes inflammation in your lungs. So some of the statistics of, So why does this virus cause this severe disease? So what is the difference between those that cause a common cold and those that can give you pneumonia? And that has to do a bit with the anatomy of the, or a lot with the anatomy of the respiratory tract. So we know that those viruses that cause the common cold preferentially infect the upper respiratory tract. And this is due to differences in the expression of receptors for the virus throughout the respiratory tract. So the viruses that infect the upper respiratory tract are going to have receptors that are commonly expressed by the cells in the trachea, in the larynx, in the nasal cavity, while those that cause disease are known to have receptors that are expressed by other types of cells that are in the lower respiratory tract. So the SARS-CoV-2 has a receptor that is known, is shared with SARS and is known to be expressed in, I think it's alveolar cells. So what's going to happen is these viruses are going to infect the alveoli, are going to cause inflammation, and are going to cause this disease, or are going to increase the severity of this disease by causing pneumonia. The temperature of the respiratory tract is also different. So you have a different temperature in the upper and lower respiratory tract, and some viruses that can proliferate very efficiently in the upper respiratory tract. Once they reach the lower respiratory tract, cannot replicate anymore. So it's probably a combination of facts. I think that there is a question. There's two questions. So let me take a look here. So one of the questions is, why doesn't it cause disease in children and young adults, and is there any precedent for this? And so that's, so actually now that prompted this question, let's talk first about the stats, and then I'll address this question. So first, the statistics. So this new coronavirus has a calculated mortality rate of 2%, but this does change with the population. So it's been estimated that it's anywhere between 0.2% to 3.4% and above, as we're going to see in the susceptible groups. This compared to SARS and MERS is lower. So SARS had around 10% mortality rate, while MERS reached 34%, which was very dire. Some of the more susceptible groups are those people with comorbidities and pre-existing conditions like cardiovascular disease, diabetes, and chronic respiratory diseases. And we also have an increased mortality rate in people 80 years and above. And now I think the estimated average age for mortality is 80 years old. But we see that the mortality rapidly increases in the group between 60 to 80. And this is the question that we had. So basically, we see that between 0 and about 20-something years old, there are not many reported fatalities. I only know of 1 or 2. And the severity of disease doesn't seem to be that bad. I honestly don't know why. It's just, for me, it was very surprising when I started seeing the distribution of these cases, and I'm probably going to dig a little bit more. So whoever asked this question, just follow up with me via email, and I will be happy. But it may also be differences in the expression of receptors, in children versus adults. It could also be differences in the immune responses between children and adults, and we know that an aging population is going to have a decreased immune response. So that could be part of that. And there is, let's see. Okay, another related question is, how does it differ from the 1918 flu, which affected mostly young people? So that's a pretty interesting question. So in 1918, we had the most severe pandemic that the world has probably faced. And it was, there's another question, but it was targeting mainly young adults. And the reason was because in this population, there was an increased immune response that would cause a cytokine storm. And most people were not dying necessarily from the underlying conditions caused by influenza, like pneumonia and others that were more common in older populations. But they were dying from the destructive effects of the exacerbated immune response in these patients. So that's why that one was particularly bad for that group. But that's not the case of what we're seeing here. What we're seeing here is that the patients that are mostly afflicted are those with debilitated immune systems. Okay. So it says news report say that people that have recovered and then show symptoms again, would it be possible to elaborate on this? So I am actually not very familiar with those reports. And as I said, many of these symptoms can be confounded with other diseases. So you have a flu, symptomatology that are very similar. You have colds, common colds that could be very similar. So I don't know if in those cases, and I'm not even know, again, I'm not familiar with these reports, but it would be very hard to say if it's due to one disease or another. Okay, any other questions? Okay, we'll move on and then I'll answer some more. Okay, so now that we said how bad it is, do we have any treatments or any options? So the short answer is, as of now, no. There are no vaccines or treatments available. However, that is not the case just for this epidemic. That is not the case just for COVID-19. In general, we have very few antivirals. We have antivirals for influenza, for herpes viruses, for HIV, and hepatitis C. This is a very limited number, so it's probably in no more than a couple dozens of antivirals that we have available right now, and that's why many of us are trying to continue our research directed in such a way that we can find targets to develop antivirals that are going to have broader spectrum capacities. But as of today, we do not have anything that is approved. However, there are some compounds that are right now in clinical trials. This is one of them, remdesivir from Gilead. So they are a company in the Bay Area that specializes in developing antivirals. So they are the ones, for example, that have susceptibility, which is the antiviral of choice that is used for treating hepatitis C patients and is actually curing these patients. This molecule is a nucleotide analog, and what's going to happen is that it gets incorporated to the RNA by the polymerase, and then replication cannot continue normally. So it's just basically going to truncate the replication of the genome, and is going to prevent the assembly, the first, of course, protein expression, but also the assembly of new infectious particles. It was not developed for this disease. It was initially developed for Ebola. It was not the most successful for Ebola, but it did show that it has other activity against other RNA viruses. So this is just one of those examples where research for one disease is probably going to give us a very good tool to target another one. So in these companies, you just don't develop a compound and then put down the drain if it didn't work for that disease. You're going to try to repurpose it. So there are clinical trials that are ongoing with these diseases I mentioned. It happened not so long ago, so they were approved on February 25, and they are in phase 3, and the reason why they are in phase 3 because some of these compounds have already been cleared for phase 1 and phase 2, and because of the urgency of these diseases, they are being expedited. It's a randomized clinical trial 1-to-1, which means that for every one patient that is going to receive the drug, there's going to be one patient that is going to receive a placebo. The administration is going to be 5 to 10 days intravenous, and there's going to be 2 studies. There are 2 studies ongoing. One is for severe patients with 400 participants estimated to be recruited, and one with mild patients where they estimate that they're going to recruit 600 participants. Although these numbers don't seem pretty high, it can be challenging to recruit this many participants, especially of the severe kind. So I don't anticipate that there will be any results in the coming weeks, but as I also mentioned, the situation is moving very quickly and people are doing a lot to try to expedite the clinical trials with these drugs. Okay, so I have, okay. I'm reading a couple of questions, so give me 1 second because over. Okay. Okay, so I'm gonna address these questions in just a moment. Let's talk about other good possibilities that we have. So besides the Gilead inhibitor, remdesivir, we have also other studies that are coming out in the last few days. So one of those is identifying the receptor or the receptors for SARS-CoV-2, and we know that we have the angiotensin converting enzyme 2, or ACE2, and the transmembrane protease serine 2. Both of these are involved in the entry of this virus into the cells, and there is proven protease inhibitor that targets the transmembrane protease serine 2, the TMPRSS2, and it basically is able to block disease in vitro very efficiently. Again, some of these is very likely that will go into the clinic or in clinical trials as soon as possible. In addition, there is an effort by the Gates Foundation, Welcome MasterCard, of $125 million to find coronavirus drugs. Some of the approaches that people take in industry and academia to try to find these drugs to expedite the process is to test FDA-approved compounds that can be easily repurposed as antivirals. And many of these studies are currently ongoing with this virus. So about vaccine development, I'm going to tell you about some alternatives that we have. So this is just a diagram with all the approaches that we have into vaccine development. So you can have different types of vaccines depending on what you use to immunize people. So you can have either a live attenuated, which is, as the name suggests, a virus that is still kind of able to replicate, but it's not going to cause disease. You can have an inactivated virus, which basically you're just developing or delivering a virus that is not going to replicate if it is dead. You can have a split inactivated, where you basically, as the name suggests, you break the virus and just deliver pieces of it. You can have synthetic peptides, so there are some proteins that are going to be more immunogenic than others, and you can just clone those proteins and use it to immunize people. You can also use virus-like particles, where there is basically a vector that you're going to deliver that has some of the proteins of the virus, but it's not going to cause any type of disease. You can do some DNA or RNA-based vaccines, again, as I mentioned, recombinant subunits. We can generate some in bacteria or viral vectors that are going to, again, have some parts of the virus, but are not going to cause any type of disease. So one of the questions that I have here is, what are the main obstacles in developing a vaccine? So I can tell you, I have a website here from the WHO that is listing all the efforts that different groups are making into trying to develop this vaccine. There's dozens of them that are using several of these strategies that I just showed you to develop the vaccine. One of the main obstacles is, well, first, depending on what you use, a live attenuated versus a synthetic peptide, what is the degree of immunogenicity and of the immune response that you're going to get to one or the other? A live attenuated is going to elicit an immune response that is going to be robust in the majority of the cases, but how long-lived is that response is going to be unclear until you test it. On the other hand, synthetic peptides may not be the best because you're not going to develop broad antibodies, but just antibodies against one or another component of the virus. And again, how good are they going to be when you're faced with a full virus is still not clear. We also have the problem of evolution. So maybe we are going to develop a vaccine that is good and the virus is going to mutate, and then the vaccine is not going to be as good. So there's a lot of unknowns. Not only that, but there is a big concern about safety. Especially with the movement of anti-vaxxers that we have throughout the world that very furiously oppose vaccines, which are going to cause a very serious problem that's going to cause even a more serious problem if we have a vaccine that's going to have secondary effects, right? And also it's production. Depending on the type of vaccine that you're going to develop, it's going to take a lot of time to manufacture something that can be distributed broadly across the world. So it's not easy. It's gonna, again, there's dozens of groups that are currently working on this, and there's coalitions of groups that are working together to try to expedite this process. But we cannot anticipate having a vaccine anytime, I would think probably in the next year. And again, for reference, we have the WHO site there that talks about vaccine development for coronavirus. So we cannot treat it, we cannot use vaccines. How are we going to prevent it? So I know that you guys have heard this, and I cannot stress this enough. I tell my kids daily, 20 times probably every day, and I'm going to tell you every time I see you, wash your hands. That's the main way. Just wash your hands constantly and for 20 seconds, and then use the hand sanitizer and don't touch your face. The other one is protect others from getting sick. If you're sick, cover your mouth. If you blow your nose, toss the tissue safely and wash your hands. Again, do not touch your face, do not scratch your eyes, do not pick your nose, do not put things in your mouth if you have not washed your hands. Prevention is going to be very tied to hygiene. These are the WHO recommendations. You can see it's avoid close contact. If you're sick, avoid spitting in public. And if you have fever, cough, or difficulty breathing, contact your primary physician and contact your healthcare provider early. Don't wait. One of the main ways that the disease can be contained is by social distancing. We can see the example in China that it did control the outbreak at some level, but it is pretty disruptive of our lives. So these are recommendations that the World Health is providing, the World Health Organization is providing. If there is a case that you have to quarantine yourself, or if there's some other measures that are taken that are more broad. This happened today, which I was kind of upset it didn't happen earlier. So this is kind of my Uber boss, Anthony Fauci, which is the head of the National Institute for Allergy and Infectious Diseases. And his statement says, coronavirus is about to change your life for a little while. This is not permanent, and there's a question about that I'm going to address. This is going to be hopefully a temporary measures. It's going to be hopefully a temporary situation. But as a community, we have to work together to try to contain, to try to prevent further spread of this virus. And the main message that I hope everybody is getting is we have to be aware, we have to be informed, but we do not need to panic. There is something that is more damaging than almost the disease is going to be the panic that this is causing. People are being inconsiderate of certain groups. Discrimination has increased. People are hoarding even toilet paper in Costco. It's getting really the panic out of control. Another statement that I really liked is from the head of the WHO, which was a couple of days ago, that the rule of the game is never give up. Never give up in trying to prevent it. Never give up in trying to understand it. Never give up in trying to control it. And I really like his words that we are not at the mercy of this virus. And we have to try to stop transmission. And that starts with us, with our hygiene. But I really like this. Let hope be the antidote to fear. Let solidarity be the antidote to blame. And let our shared humanity be the antidote to our shared threat. And we are not at the mercy of this virus, which is something that we have to remember. With that, I'm going to close and then I'm going to take some questions, but I want to leave you with this message that viruses are everywhere. So this is a pandemic that we are unfortunately facing at this moment, but it may not be the last one. It certainly is not the first one. The more we understand about the biology of these viruses, this is going to bring us closer to treatments and vaccines, and even to understand more about nature and ourselves. And we, as a community of scientists, we're putting a lot of effort in trying to understand, again, the origins of this epidemic, the nature of the virus, and the nature of disease. And with that, I thank you all for the effort of being here remotely. It's pretty good. I'm gonna take some questions, and just remember the message there. Keep calm and wash your hands. And now I'll take some questions here. Okay. And you guys, if you have more questions, I have about 4 questions that I'm gonna answer. If you have more, you can always send it via chat. So one of the questions is if COVID-19 is likely to pass or go away like SARS and H1N1. It is possible. So we saw with the SARS epidemic that after a while it dwindled and it was under control. And as I said, there are sporadic cases, but none of them at the same level of what we saw in the first pandemic. Again, the infection pattern of this COVID-19 is different. So it's still, we're going to have to determine exactly what is going to be the outcome of this virus. So we still don't know, but at least from what we know of other coronavirus epidemics, it's possible that it will. Okay, so good. So there's a question that is related with that, which is if the virus can remain latent in the body after the symptoms subside. So hold on a second. So I actually don't know the answer to that, and from what it looks like from the patients that have recovered, it is unlikely, but I'm not sure of what measures have been taken to really study if there is any viruses that remain. From the other epidemics and the other viruses, it doesn't seem like these viruses are going to behave like a herpes virus that is going to be with you forever, but it seems like you are actually going to get rid of this virus after the disease state has passed. That said, I'm sure that there's a lot of investigations going on. How long do you have the virus, or how long are you transmitting the virus after you don't have any more symptoms? Okay, let's see. So this is also an interesting question. So how are fatality rates of pre-existing conditions correlate with those as a function of age? So we do know that in aging populations, these comorbidities are going to increase. So it is very likely that in this population, we have more people that are going to be prone to be diagnosed with cardiovascular diseases or diabetes, just as age-related again, comorbidities. So it is possible that both of those are linked to each other. That said, we do know that the aging immune system is not going to be able to handle disease as well, and it's also going to contribute to the severe cases that are developing. So there's been a lot of talk about, hold on. I'm gonna get my facts straight here. So there's been a lot of talk about zinc lozenges and coronavirus, and I must confess that if there is, I don't know what the mechanism is. It's something that I probably will look into this one on vitamin C, but that's again, email me and I will be happy to answer that. And that the other question that we're having is what do we know about the SARS-CoV, the COVID-19 tolerance to temperature? So this comes from the question of, is this epidemic really going to dwindle down in the summertime? And the answer is we don't know. It's not well understood even for influenza, really what are the effects of temperature. So it has to do a little bit transmissibility, for example. So the droplets, the way that they travel and how far they travel is going to change with temperature. But I don't think that is that the virus is directly killed, or it's just different. Also, the environment in a dry winter is going to be the environment of your mucosa is going to be very different in a dry winter than in a moist summer. So that's also going to help more with transmissibility. So hold on, let's see, find this. So those are all the questions that I have here. And if anybody has any more questions, just feel free to email me, and I'll try to answer them in a timely manner as you can. Oh, okay, I think there's 2 more questions. Hold on. And I think we have 2 more minutes, so I'll wait. But I will try to answer the questions that you have in a timely manner. It is a pretty busy and hectic time, as many of you can imagine, but I will definitely do my best on trying to answer some of these questions. Let me see if we have these here. Also, one question here is how are tests conducted? So the majority of the tests that I'm aware we have are going to be PCR based. They are going to, that was also one limitation. So of how could we discern from circulating strains from those circulating strains and the COVID-19. So the specificity of the tests was a challenge, but I think finally they are good, and they are being the majority of what I know is by PCR. Okay. One more. Okay. That was a long one. Okay, I'm gonna take this time to acknowledge two of my secretaries, Dr. Matthew Lewis and Dr. Diego Acosta, who are wonderful at typing out the questions that you guys have. Okay, so the question is, in a population with stringent isolated protocols in place to limit transmission, what criteria will be used when these protocols would be eased? That's a good question, and I think these the protocols are constantly evolving, but they're going to have to do with likely the number of cases that are diagnosed in a community. How are the exposure measures being controlled, but especially how are you limiting the expansion of the epidemic? So it's likely going to do with just how many cases do we have in a particular geographic region, and also on how we are testing and how efficient are those tests. Okay, anything else?
I think that's it.
Okay,